through the steps of how to perform a urinalysis, okay? So today we actually have 2 samples, and with that we have Corny's sample, which is going to be in our sterile cup. This is a free catch sample, and so we have it dated and we have a time. So when we get a urine sample, it's important to put the date and time on it, because we know that we run a urinalysis, we want to make sure that we have a sample that's within an hour's time, okay? Any urine sample that we collect, say a client brings it in, and if we can't run that urine sample right away, it needs to be refrigerated. Any type of bodily fluid or sample that sits out in regular room temperature, then it's going to start producing, or bacteria is going to start to multiply in it. So with that, I'm going to demonstrate with Corny's free catch sample, okay? Remember other ways that we can obtain a urine sample. If we need a sterile sample, we're gonna catch that by, or collect that by a cystocentesis. Or if we cannot get, say it's a male dog and they will not urinate, we will do a urinary catheterization, and that's another way that we can get a sterile sample. So, for demonstration purposes today, we're going to start off with, we want to evaluate and we want to do a visual observation of the urine. So this is important. So when we look at Corny's urine, her urine is very gold, dark gold in color, okay? It's got a little bit of cloudiness to it. So that's going to be the turbidity. So we're looking at the clarity, how clear it is, and we're also looking at how cloudy it is. That's going to be the turbidity of it. Obviously, when you take the cap off, you're going to smell it, okay? So some urine samples are going to be really strong, and that's pretty obvious. So with that, it's not like we're expecting to put your nose down there, but as you open it, you're going to be able to smell. The main things that we're looking for is the turbidity. We're looking at the color, okay, and the appearance of the urine sample. So that's what we're going to do right offhand. So from here, I'm going to take my sample, and then I'm going to transfer it over into either my centrifuge tube. For today's purposes, I'm just going to be using a regular red top tube because this is a sterile tube, okay? But we can also use your sterile centrifuge tube. So for urine collection, depending upon your practice, typically all we need is 1 cc of sample. So you can go up to 1 ml to 3 cc's of urine, okay? Some actual practices have little tiny microtainer, the little tiny tubes that we can actually spin in a little stat spin centrifuge. We don't have one of those, so we're gonna have to use these, okay? So you're gonna take the top off, and then you're gonna pull up your urine sample. So for today, we're gonna pull up 3 cc's of urine, and then you're just going to place that right into your sterile tube. You guys will have an actual urinalysis form that you will be filling out this information as you're getting it, okay? So I do not have that right now. So this would be our sample, and then obviously we would then go, and this sample would be labeled as well, okay? So with that, you would take your Sharpie, and then you would write the animal's name and last name on that sample. So the next part that we're going to do is going to be your urine dipstick. Thank you. Your urine dipstick and your specific gravity. So this is Bella. No, this is Corny. Okay, so you have your sample. So what I'm going to do is I'm going to use my 1 ml syringe. I'm going to go into my sample, and all you need is 1 drop. So I'm going to place that on my refractometer. Your refractometer is an instrument that's going to measure specific gravity or the concentration of the urine. So what you're going to do is you're going to hold it up to the light, and where the blue and the white line meet, you're going to take your reading. And right now, this specific gravity is at 1050. So you're going to look at the column all the way to the right of this particular refractometer. It's 1050. So when you take to clean this, you're going to use either run it under the water and use a soft towel to clean it, and then place it back into its case. Do not use anything that can actually scratch it, like a Scotch-Brite. Okay? Excellent. All right, the next thing you're gonna use is your urine dipsticks. So that's gonna be your next step. Okay, whoops, that's a new one. Let me grab one that's open. So your urine dipstick, you're gonna take and pull that out. And some people will actually take, if you have extra urine, you can just take your syringe and then just go across your little squares and place the urine on top. Okay? And if you look on, or when you guys see your bottle, it'll actually have on the side the time. You're supposed to wait about a minute or 60 seconds. Okay? Remembering that when we're running a dipstick, we're looking for a color change. That's what we're looking for. Okay? So with that, we're going to wait about a minute, and then we're going to go to our bottle. Remember that you are actually adjusting your urine dipstick to where it's located onto the actual results. So at the bottom or the top, it'll have a little square that says place the handle here. Okay? So all you have to do is you will just go along, make sure that it matches up, and you want to go right along, like where it says glucose. See how blue it is? That means it's negative. Okay? We have protein, it's a bright green in color, and we're going to try to match that the best you can. That's at 1+, so that's not something we're going to be too concerned about. Your leukocytes, they're negative. Another one we're looking at is bilirubin. So you guys talked about that. We're looking for a color change, and because the urine was so dark, it's hard for us to look at that, but really none of those colors match up. Okay? So as you guys are taking those results, you're going to be putting those on your urinalysis report sheet. Okay? Any questions? Yes, ma'am.
What happens if they don't match up? Like, what does that mean?
If they do not match up, you're going to go to the color that comes as close to it as possible. A lot of times if it doesn't match up, it's because your urine sample has blood in it, okay? Or the bilirubin is so high that it's going to skew the color of it. Okay? All right. So now what we're going to do is we're actually going to have to take this sample and spin it down. So this is going to have to go into the centrifuge. All right? So I'm going to say your machine, in that particular machine, you need to make sure that your centrifuge is turned to urine. Okay? Our centrifuge has actual numbers on the front where Jacob is that gives you the actual where you're going to change it from blood to urine to feces. Okay? I currently have changed it to what you're going to run this on, urine, in that particular centrifuge. Okay? All right, so let's pretend that I've already spun this. Okay? After you pull it out of the centrifuge, what's going to settle at the bottom? Sediment. Okay? So that's why it's called your sediment evaluation. So what's going to happen after you spin it? The sediment, or what we call a plug, is going to be at the bottom of your tube. That is where all your cells are going to be. Okay? The top of that is called the supernatant. That's the term for it. So the liquid portion at the top is called supernatant. What you're going to do is you're going to come over here, or in the sink. We're going to be in a lab with a sink, and you're going to pour off your supernatant. Okay? And then what you're going to have, and we haven't spun this down yet, but you're going to have a little bit of your sediment or your plug at the bottom. Okay? So the first thing we want to do is we want to go ahead and get our sample for our urine cytology. So what I do with that is I'm going to take my syringe, pull up a little bit of my urine sample, okay? And then I'm going to put a little bit on the microscope slide, okay? So I'm going to take and put a drop, and then I'm just going to let it run down the slide, okay? If we have a plug that's really thick and looks real cloudy, that may mean that we've got a lot of cells in it. So that's when I like to do a urine cytology, because I can see a lot of cellular matter that I will not see on my urine sediment. So we would place this here to allow it to dry, okay? Then from there, you're going to take your next bit from your sediment, and you're going to place 1 drop of urine with the cover slip. A lot of your technicians now are trained to look at unstained samples of your sediment. I am old school, so I personally like to look at it with SETI stain, because that's the way I was trained in school. This is called SETI stain. It's purple, okay? So we're gonna take the top off. We're going to put just a little bit. It's gonna depend on your sample, okay? Remember, it doesn't take a whole lot, so we're just gonna do a little drop, okay? I'm gonna put the top on, and then I'm just gonna roll it so it mixes, just like that, okay? So it's evenly distributed. After that, I'm gonna take my syringe, go down into my sample, and just pull out enough for a drop. I'm gonna place that onto my slide. And then I'm going to place my cover slip right on top, okay? So you have your unstained, and I have my stained, okay? So you guys as veterinary assistants, you will find out what your technician's preference is, or go ahead and make both if you're unsure, okay? Once we make this, you're gonna put it over, so this would go over to the microscope, okay? And then you would then place it on the stage, or you would then let your technician know that your sample is prepared. When we are looking at our sample under the microscope, we're gonna put it on 10 power first with a low light. So we're gonna turn our light down. If not, it will burn through the cells and you cannot see them. So we're gonna get it into magnification, and then we can go all the way up to 40. You do not go to 100, okay, with the urinalysis. So 10 to 40. Okay? Your cytology. So what you're gonna do with your cytology, once the sample is dry, okay, we are now gonna diff quick this particular sample. Again, we do not want to use the same diff quick stain that we're using for normal cytologies, okay? And we do not typically want to use it for, so like fecal, okay? We have to be careful because of bacterial contamination. So with that, make sure that you're not using it with the same stain that we're using for cytologies, like fine needle aspirates or blood smears or any other so. With that, we're gonna start with our diff quick stain. This is a 3-step process, okay? So first step, you're going to place your microscope slide in each step of your stain for 1 minute. You start with your blue, red, final step is the purple, okay? So you're gonna keep that in there for 1 minute. Once you're done with the purple, okay, you're going to take your purple and you've gotta wash it off. So after you're done, it's done for 1 minute, you are going to go over and then you're going to actually run this under water, take a blow dryer, you're going to dry it, and then you would come to your technician and say, Mrs. Small, I've got your urine cytology prepared, okay? Another good habit to get into is to actually label the top. This is frosted, so use a pencil or a Sharpie, and you would write corny, call, okay, on your actual slide, and then that way it's identified. Okay, so that is your urine. When we're starting with our visual, our dipstick, specific gravity, centrifuge. Then from there, we're going to pour off our supernatant. So we're going to prepare our sediment. Then we're going to take a sample with unstained with a cover slip, drop of SETI stain, cover slip. And we're going to let your technician look at that. And then you've got your urine cytology that we will evaluate. On the urine cytology, that's where we're looking for bacteria, white blood cells, epithelial cells, and we're also looking for those neoplastic cells that we talked about in your groups today. That can be a result from the kidney or from the bladder. Your urine sample, once you are done, okay, please make sure that you check with your doctor, because for any reason, the doctor may want to see the sample, okay? Or they may say, hey, I'd like to get a culture and sensitivity from that. So you may want to double check with your doctor before you throw out or discard your sample. Okay? The other thing that I do want to add into for catching your sample, we use a product called Nosorb, and that's for cats. So if I have a cat that basically we are trying to get a urine sample and say they're not very nice and I can't express it and we can't get a cysto, the Nosorb is exactly what that means. It will not absorb the urine, but it gives them a substrate that they can actually scratch in. They urinate in the litter pan, and then we just do a free catch sample directly out of that, okay? So when you guys hear about Nosorb, or make sure you go and give the owner some Nosorb, that is what we're talking about. And they're just little plastic beads. We put them in a little cup, and we reuse those routinely in practice. That concludes our demonstration of how to perform a urinalysis. Thank you.